Hello, I'm Katie Cottingham, and welcome to this news briefing from the 246th National Meeting and Exposition of the American Chemical Society in Indianapolis. We're joined today by Dr. Rahman from Applied Research and Photonics, who will give us a brief overview of his research. For those of you joining us here in person, please silence your cell phones, and when asking a question, please state your name and affiliation using the microphone that we've provided. We're also joined by reporters online via our ACS Live Ustream channel. All reporters are encouraged to ask questions after the researcher gives his overview. Also joining us are ACS science writers who might also have questions, but we will field those from news media and bloggers first. For online media, we ask that you embed or link to the archived version of this session, which will be available at www.ustream.tv/channel/ACS Live. So now we will turn to Dr. Rahman, who will talk to us today about T-rays, which offer the potential for an earlier diagnosis of melanoma. Dr. Rahman?
Thank you for the invitation and for the opportunity to speak with you. T-ray stands for terahertz radiation. So it is the portion of the electromagnetic spectrum that's in between infrared and microwave radiation. So there's something special about this portion of the energy on the spectrum. The radiation is a general name, of course, so the ray is an abbreviation. T-rays are non-ionizing, but it can penetrate through certain things, or most of the things except metal. So that includes the human skin and other tissue, as well as many other substrates. For example, box inspection, etc., also can be done with terahertz. So in case of diagnostics on human in vivo, which is the next step of our goal, so far we have been doing a lot of in vitro studies, particularly on stratum corneum, with very limited experiment on a live volunteer as well. But the property that we want to utilize here is the fact that we can non-destructively and non-invasively focus the energy under the skin and then study if there is anything abnormal over there. And this can be done one of the two ways, probably a combination of both ways most of the times, that we can either take an image of the suspect area, not only on the skin surface, but also, as I said, under the skin surface at different depth, to see if there is anything that looks totally different than the normal healthy tissue. And then also, because it is focused, from that point, we can take spectrum. And once we take the spectrum gives us a lot more detailed story, but it's also not so easy to interpret. A lot of times it's because a lot of information are coming out. Nonetheless, if we can generate a library of the healthy tissue of different organs, then comparing to that library, we can come up with a protocol or procedure arriving at whether this is melanoma or this is something else or some other abnormalities. So some work need to be done, nonetheless, because we can see under the skin non-destructively. It's very important and attractive way of coming up with a new diagnostic tool. And we're still working on it. Our instrument is now taking images under the surface, but going to in vivo will take some more modification.
Okay, so do we have any questions? Kath?
Kath O'Driscoll from Chemistry and Industry magazine. I would just like to know, what are the molecular changes that you see in skin, particularly sort of in the area of cancer? What kind of results have you been getting?
Well, this is actually pretty complex question. So if you're looking on a cellular level, for example, which is probably the most effective diagnostic tool, then you have to be able to compare a healthy cell with a non-healthy one and establish what kind of non-healthiness is there. It can be melanoma, but it can be something else. So the false alarm of melanoma should be reduced, and a signature need to be established. But a cell by itself is a very, very complex system. There are so many things inside a cell, including the most attractive one is the DNA, the chromosomes. And a particular segment of the DNA is the genes. So on another scale, we are also doing that research in collaboration with Penn State College of Medicine. So if we are able to now discern not only a cell response, but also the component or composition elements of the cell's response, that would be even more effective. And this also, in this connection, I think I should mention that J. Michael Bishop, Nobel laureate, wrote on genetics of medicine and so on and so forth, mentioned that the journey of a cell from being healthy to non-healthy, such as cancerous, is a long process. It does not happen overnight. And there are self-correction mechanisms within cells, and when that mechanism fails, only then the cell converts into a cancerous cell. And understand that our immune system is supposed to prohibit that kind of alteration or take out from the system. Nonetheless, these changes happen on such a small increment that our immune system keeps thinking that this is normal, and there is no check and balance there, and we get tumor all of a sudden. Nonetheless, this process is very, very lengthy process. So if there was a way we could understand that a cell is now doing healthy function, and all of a sudden it started moving towards the non-healthy functionality, that mechanism is not understood yet. If we can understand that mechanism, that also give us a wealth of information towards the cure, towards finding cure.
So how early in that process of cancer formation could you actually detect a cancer? I mean, how much before you actually might see other symptoms manifesting, could you actually detect these composition changes perhaps that you're talking about?
Well, that is the idea, but we are not able to do that yet now. The current detection system can only detect when one of the four phases are there at the earliest phase, the first phase, second phase, third phase, and metastasis. I forget the term, the fourth phase of the cancer. But it has to be in the first phase for the current technology to be able to detect. That's not a molecular level detection, not even, let alone a genetic level detection. So some work need to be done so that we can, at suspected sites, we can take a spectrum and make a prediction that if this thing is going to get into a cancer or not. Some work need to be done. At this point, I don't think we have any capability of doing that.
Okay, thank you.
Okay, Doug?
Doug Dollimore, Science Elements Podcast. Doctor, what led you to start investigating this use for T-rays, and what sort of adaptations did you need to make to make it work for detecting melanoma as opposed to, finding the gun underneath somebody's jacket at an airport?
Right. As diverse as different as those two applications are, as you can imagine, with T-ray it is related actually. So we started. We founded this company, Applied Research and Photonics, in 2003, with a slightly different goal, and in keep doing research with nanomaterial called dendrimer, we were able to make a high-power CW source, which is slightly different than all the other technology available for generating terahertz. So, and then we realized that it has a number of important applications. Imaging being one of the main attractive application, because you can see under the surface, under something, behind something, without breaking it, without destroying it. So obviously we started thinking that it should be able to become a diagnostic tool such as melanoma, but it may also alternative of mammogram and that kind of detection tool it can be. And if the spectroscopy, a simple analogy is in the spectroscopy that when you go to your doctor, they put the probe on your heart and you see the pulse, you have no doubt that there's a beating heart. The same stage of development need to be done, that you take a spectrum from some portion of your body and you can predict that that's a disease or that's something else. And all the bacteria, all the pathogen that gets in our body or disease conditions, each of the molecules have their own unique signature. And we generate terahertz by electro-optic dendrimer over a very wide range, up to 35 terahertz. So within this range, it's like a big window of observation. Within this window of observation, every molecule is different. You can see clearly the difference between two molecules. If you are able to do that, then it should be possible to get into the diagnostics. But by the same token, as you had mentioned, finding the gun under somebody's shirt. So in that case, you're not looking so closely on a molecule, you're looking on a macroscopic level, which also we have demonstrated, by the way, that you take a broader picture, such as they are working on terahertz camera, which is still on the way, but we have been able to use high sensitivity IR camera, and still, because IR is the infrared and terahertz overlaps to far IR, far infrared, and so that is uncharacterized region of those IR camera. And we have been able to use that to see metal form and stuff like that under the shirt or under inside a box. Now, what adaptation we need to make it a diagnostic tool? Well, first of all, unless you have funding sources, you can do nothing. We being solely in the private sector, depending on a small revenue that we are generating by providing services, and also we are having some angel investors investment. That's how we are doing it. And if we are fortunate to get enough funding, then we could partner with physicians who are the point of service person to this kind of patients. And that interaction is very, very important. And we need that to be able to come up with a tool that will be actually used for diagnostic purposes. So those are the hang-ups that we have.
Okay, other questions? Kath?
So I just wonder if you can say a bit more about the in vivo, the plans to do in vivo work. What will that entail exactly?
You mean what we have done so far, or what we could do?
What your next stage is, yeah. Because I thought you said you'd worked on stratum corneum. Is it originally?
Yeah, right. So as you know, the drug delivery is an issue. The transdermal process is much, much simpler compared to injection or swallowing a pill. You can put active straight into your bloodstream. If it can go through your skin or stratum corneum. Now, in doing so, they need to know some crucial information. One is it actually penetrating through the skin or not? And the second is how fast it is. If it is going there, then how fast it is going. And third is how much of it is going. And the way there is no non-invasive way of doing this kind of measurements, except the mechanism that we have come up with. So we can directly measure if I put something on my skin, then I can quantify it because I can now scan across the depth of my skin. So I can directly measure what is the concentration distribution across the thickness of my skin of that particular analyte that I have just applied. So these are the, this is ongoing, but it's not very invigorated because shortage of funding and partner. So that's what we need to do next.
I would imagine that's quite useful as well for cosmetics, is it? Because a lot of cosmetics are talking about penetration across skin as well. Where I thought there would be more money.
Well, the cosmetics or personal care products in general, they are one of our target market sector, and they have direct use of this technology. So it's not in their case, it's an adaptation. It's a diffusion of diffusion of knowledge, so to speak. Because the lab people who are doing the actual testing for personal care industry, we are working with one company right now. They are very much interested to adapt our technology in their research and development plus quality control. But it's a matter of, for them, they know that there is nothing exists that they can use right away. And we have not been out there so much educating them that they can do what kind of direct measurement with us.
And just a sort of last question to ask, how do you envisage this might be used in a future scenario for actual sort of disease detection? Would it be like an airport scanner? Would we have a scan in order to detect certain diseases using this?
It would be some kind of a scan, that's for sure, except that it's a much simpler way of scanning. Of course, we're not thinking about whole body scanning like MRI. We are, for melanoma, it's a very isolated location, so you scan only that particular location in a non-contact fashion. So there is, the radiation is reflected off of that surface, and because we can selectively focus it on a given layer, so then you do the layer by layer scanning to interrogate the situation. So yes, it's a scanner, but also it could be a spectrometer. You can take the spectrum, as I mentioned already. And the image, of course, image is formed. This imaging is called, what they call is a reconstructive imaging. So that's always, as you scan, you have the data, and you convert that into image.
Right, thank you.
Okay, one question in the back. Last question.
Christine Sa, ACS News Service. Would it be possible, in theory, to create a device that uses T-rays, let's say for a surgeon who's removing a cancerous tumor on the inside of a patient, to have a handheld device to, once they remove the, what's visible of the tumor, to see the lining of the other parts of the body nearby and make sure that they remove all of the cancerous cells.
Right. So that's another interesting area. So now the what they call is a tomography, intravenous or interartery tomography that's being done. So what you do is you put a fiber in there, reflected light is collected, and the image is created. It's being done with near IR radiation right now. But as you have mentioned, that can you see then what is inside the artery wall, or what is inside the tumor or in the associated area? That would be facilitated by using terahertz radiation. So that's another important area that we could explore, or anyone could explore. I know NIH has probably some program, but unfortunately we are not associated with that. We wish we were. We wish we could, because we have the most useful terahertz source. We also have a sort of a plan and design to use with the fiber. So fiber coupled device, actually we have a small demonstration in our lab that we can show the terahertz is being coupled to the fiber, and then bringing it back and making the image just like any other tomography system. So that's, of course, an important area, and we could get into that had we had the opportunity.
Okay. Thank you very much for joining us, Dr. Rahman, and please stick around for our next press conference, which is on the symposium Biopesticides, State of the Art and Future Opportunities. Thanks a lot.
Thank you for having me.